From the smallest single-cell organism to the largest creature on Earth, every living thing defined by its genes. The DNA contained in our genes acts like an instruction manual for our cell. 4 building block code bases are strung together in precise sequences. We tell the cell how to behave and form the basis for our very trait. But with recent advancement in gene editing tools, scientists can change an organism's fundamental feature in record time. They can engineer drought-resistant crops and create apples that turn brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where does this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a lateral process that long-fashion air bacterial immune system, regularly found defending single-cell bacteria and a cure against inviting viruses. Naturally occurring CRISPR use 2 main components. First are short snippets of repetitive DNA sequences called clusters regularly interspaced short palindromic, or simply CRISPR. The second are Cas or CRISPR-associated protein, which throw up DNA like molecular threads. When a virus invades a bacterium, Cas protein cuts out a segment of the viral DNA to stick into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. The viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a precursor protein called Cas9. The resulting complexes act like scouts, lodging on the free-floating genetic material, searching for a match to the virus. If the virus invades again, the scout complex recognizes immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target larger viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool. We can alter DNA and gene-specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene that they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein molecular scissors slip the DNA. This is the key to CRISPR power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is caught, the cell will try to repair it. Technically, protein cause nucleases trim to broken ends and join them back together. But the type repair process, called non-homologous joining, is prone to mistake and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular protein can perform a different DNA repair process called homology-directed repair. Template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even research a completely new one. Ability to fix DNA in raw means that CRISPR could potentially create new treatment for diseases into specific genetic in raw, like cystic fibrosis or sickle cell anemia, and since it's not limited to humans, with application on almost animals. CRISPR could create plants that dry larger fruits, mosquitoes that can transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying in general, allows scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make choices in intended changes, and since it's difficult to predict the long-term implications of a CRISPR edit, the technology raises big ethical questions. It's up to us to decide the best course forward, and CRISPR leaves single-cell organisms behind and heads into lab, farm, hospital, and organisms around the world.